Wnt glycoproteins are signaling molecules that control a wide range of developmental processes in organisms ranging from the simple metazoan hydra to vertebrates. The Wnt or wingless signaling network regulates diverse processes during development such as cell fate determination, structural remodeling, cell polarity and morphology, cell adhesion, migration, proliferation, and growth. The network is composed of secreted glycoproteins, Wnt family ligands, and the frizzled and LDL receptor-related protein LRP families of receptors. In general, there are three signaling pathways associated with Wnt receptor interaction. The first is commonly called the canonical pathway. The heart of this pathway centers on beta-catenin. This pathway can be either active or inactive. I will now demonstrate how the canonical Wnt pathway works by using assorted kinds of candy. In the activated pathway, the canonical Wnt signaling pathway is induced when the Wnt ligand binds to cell surface receptors of frizzled and the co-receptor LRP, causing activation of Devel. Proteins make up the destruction complex. These proteins include axin, beta-catenin, APC, CK1-alpha, GSK3-beta, and others. Disheveled induces dissociation of the destruction complex. This causes accumulation of beta-catenin in the cytoplasm. Beta-catenin is then free to enter the nucleus and activates Wnt-regulated genes through its interaction with co-transcription factors TCF and LEF. TCF and LEF itself activates an RNA polymerase which induces gene transcription. Examples of important genes induced by the active Wnt pathway are genes coding for c-Myc and cyclin D1. Normally, in most cells, the Wnt pathway is inactivated. Wnt does not bind to frizzled, Devel is not activated, and the destruction complex does not get shut down. The ubiquitin ligase beta-TRCP is able to bind to beta-catenin and able to transfer short ubiquitin peptides to bind to beta-catenin and direct it to the proteasome. Ubiquitinated beta-catenin is bound and destroyed by the proteasome, which is called proteasomal degradation. In the nucleus, TCF and LEF, together with other proteins, bind to DNA and inhibits gene expression without beta-catenin.